Hello everyone and Assalamualaikum. In this video, I am going to talk about Introduction to Gastrointestinal Function and Physiology of Smooth Muscle. First, let us look into the functions of the gastrointestinal tract. One of the functions of the gastrointestinal tract is for ingestion. Ingestion involves taking of food and fluid into the mouth. Another function of the GIT is secretion. Water, acid, electrolytes, and enzymes are secreted into the GI tract in order to facilitate digestion. The GI tract also performs the digestion process itself. There are two types of digestion, which are mechanical and chemical digestion. Mechanical digestion is performed by teeth, which is the process of chewing or mastication by the teeth. During chewing, food is manipulated by the tongue, ground by the teeth, and mixed with saliva to dissolve it. As a result, the food is reduced to a soft, flexible, and easy to be swallowed, called bolus. After passing through the stomach, the bolus again will undergo another mechanical digestion by the smooth muscle of stomach and small intestine. The product of digestion is called chyme. In the intestine, motility of the small intestine will mix the chyme with digestive enzymes and pancreatic secretions. Another type of digestion is chemical digestion. In chemical digestion, complex food molecules such as carbohydrate, lipid, protein, and nucleic acids are split into smaller molecules by hydrolysis. This process is catalyzed by digestive enzymes. For example, in the mouth, salivary amylase acts on ingested disaccharides and starch to be broken down into monosaccharides. Hydrochloric acid activates pepsinogen into enzyme pepsin, which then helps digestion by breaking the bonds linking amino acids, a process known as proteolysis. GI tract also functions for mixing, which is the contractions and relaxations of the GIT muscle to mix food with secretions. GIT also performs propulsion, which is to propel or to push forward the digested food along the length of the GIT towards the anus. Another important function of GIT is absorption. Water, vitamins, and digested substances such as glucose, amino acids, small proteins, and triglycerides are absorbed from the lumen of the GIT into the blood and lymph vessel and then be circulated to cells throughout the body. And finally, another GIT function is defecation. The GIT eliminates unwanted materials such as waste, indigestible substances, bacteria, and dead cells that is sloughed from the lining of the GIT. These unwanted materials is termed feces. So basically, these are the major functions of the GIT. Now, let us look into the physiology of smooth muscle. First, let us look into the locations of smooth muscle in the GI tract. The mouth and pharynx actually consist of skeletal muscle, which function to help swallowing. The rest of the GI tract consists of smooth muscle, 3 layers in the stomach, and 2 layers in the small and large intestine. This diagram shows cross-section of small intestine. Here we can see the wall of the intestine consists of 2 layers of smooth muscle, which are longitudinal layer, the outer layer, as well as circular layer, the inner layer. So the longitudinal layer contracts, relaxes, shortens, and lengthens the gut, allowing food to move in one direction. The circular layer allows the gut to contract as well as to break large food particles apart. The circular layer also stops food from moving to the wrong direction by blocking the more proximal end. So the two smooth muscle layers work together to propel food from the proximal end to the distal end. In some regions, the circular layer of smooth muscle enlarge to form sphincters. Sphincters are circular muscles that control the opening and closing of the lumen. For example, between the stomach and small intestine, as well as between small and large intestine. There are three types of muscular tissue: skeletal, cardiac, and smooth muscle. These three types of muscle differ in their location, function, appearance, as well as control. Skeletal muscle is mostly attached to skeleton. Cardiac muscles forms the heart, whereas smooth muscle can be found at various places in our body, such as the GI tract, uterus, in the eyes, and blood vessels. In terms of function, skeletal muscle is responsible for body movement as well as posture. Cardiac muscle is responsible for keeping the heart pumping and blood circulating around the body, whereas smooth muscle perform various functions such as peristalsis, oval movements, control pupil size, and control blood pressure in the blood vessels. In terms of appearance, skeletal muscle is striated and multinucleated, meaning there is more than one nucleus. Cardiac muscle is also striated, but it has only one central nucleus, whereas smooth muscle also has one central nucleus, but it is non-striated. The control of skeletal muscle is voluntary, whereas cardiac and smooth muscle are involuntary, meaning that they are controlled by the autonomic nervous system. So those are the difference between the three types of muscular tissue. For this lecture, let us focus on smooth muscle. Smooth muscle structure differs significantly from that of skeletal muscle. This is skeletal muscle and this is smooth muscle. Its most striking feature is the lack of visible cross striations, so that's why the name is smooth. Smooth muscle also composed of smaller fibers, usually 1 to 5 micrometer in diameter and only 20 to 500 micrometers in length. Skeletal muscle fibers are about 30 times greater in diameter and hundreds times longer. This figure shows the structure of smooth muscle. This is a structure of one individual contractile unit. Smooth muscle contains actin that is attached to the dense bodies, and myosin filaments that are not organized in sarcomeres. A sarcomere is the basic unit of striated muscle tissue, which is the repeating unit between two Z lines. Sarcomere are the one responsible for this striated appearance. Since the actin and myosin of the smooth muscle are not organized in sarcomere, this gives the smooth muscle the non-striated appearance. Another difference between smooth and skeletal muscle is smooth muscle does not have transverse tubules or T-tubules. In skeletal muscle, T-tubule conduct. This is the T-tubules in skeletal muscle. T-tubule conduct impulse from sarcolemma into the cells, and the sarcoplasmic reticulum stores calcium ions. Smooth muscle does not have these T-tubules, but instead it has caveolae, which is the invagination of sarcolemma membrane. This caveolae contains extracellular calcium. Smooth muscle also has fewer mitochondria and sarcoplasmic reticulum. There are two major types of smooth muscle, which are unitary, or also called visceral or syncytial, and multi-unit. The axons that innervate the smooth muscle do not have the typical branching end like in the motor end plate of skeletal muscle, but the terminal axons have varicosities. These varicosities are filled with neurotransmitters such as acetylcholine, norepinephrine, and perhaps other substances as well. So let us look at the difference between these two types of smooth muscle. Unitary smooth muscle consists of many smooth muscle fibers that contract together as a single unit. The fibers are arranged in sheets or bundles, and there are gap junctions between the cells. So because of these characteristics, it allows action potential and ion flow throughout the organ to be faster. So that is the feature that makes them able to contract together as a single unit, meaning it performs one function together. Another special feature of smooth muscle is that it can contract when they are stretched, even without extrinsic innervations, either sympathetic or parasympathetic. For example, in the GIT, presence of food in the stomach will cause stomach muscle to stretch, and this will stimulate the muscle to contract. When the smooth muscle is stretched, membrane potential becomes more positive, which leads to an increase in the frequency of action potential and subsequent increase in muscle tone. So where can the unitary smooth muscle be found? It can be found in the walls of hollow organs, such as the GI tract, uterus, bladder, bile ducts, and many blood vessels. Because it is mostly found in the walls of organs, this unitary smooth muscle is also called visceral smooth muscle. Multi-unit smooth muscle composed of individual separate smooth muscle fiber. It is densely innervated by post-ganglionic fibers of sympathetic and parasympathetic nervous system. Each fiber is often innervated by a single nerve ending, so it can contract independently. There are little or no gap junctions between the cells, so that is why it can produce fine, localized and graded contraction, meaning the contraction is very specific at one very particular location. So, where does the multi-unit smooth muscle can be found? Some examples are iris, ciliary muscle in the eyes, bronchioles, piloerector muscles, and vas deferens. Again, to differentiate between the unitary and multi-unit smooth muscle, the unitary smooth muscle, such as in the intestine, when there is action potential, neurotransmitter will be released from the varicosities into the cells and through the gap junctions. So these several smooth muscle cells will contract together as a single unit. In multi-unit smooth muscle, such as in the eye, each muscle fiber is innervated by sympathetic and parasympathetic nerve endings. There is no gap junctions, so each fiber will function independently. There is also places we can find a combination of unitary and multi-unit smooth muscle, such as in the blood vessels. There are various functions of smooth muscle in the body because it is found in various locations. For example, in cardiovascular system, it maintains the tension in the walls of blood vessels in order to regulate blood pressure and blood flow. In respiratory system, contraction and relaxation of smooth muscle will regulate the inflow and outflow of air. In urinary or renal, smooth muscle function to regulate renal blood flow and glomerular filtration. The smooth muscle of the ureter helps to propel urine from kidneys to urinary bladder. In reproductive system, it facilitates the movement of sperms and accelerate the movement of ovum into uterus through fallopian tube. In GI system, smooth muscles such as in the intestinal wall helps in the movement of food substances mixing with digestive enzyme. In large intestine, it functions to facilitate defecation. Excitation-contraction coupling. Excitation is the production of the electrical impulse, which leads to contraction. Hence, the name is excitation-contraction coupling. So, excitation-contraction coupling in the smooth muscle is slightly different than in skeletal muscle. Nerve stimulation will cause membrane depolarization in both skeletal and smooth muscle. So, both in skeletal and smooth, membrane depolarization will occur when there is nerve stimulation. But in smooth muscle, the binding of actin and myosin happens when calcium binds to calmodulin, which is a calcium binding protein. In skeletal muscle, calcium ion will bind to troponin C, which causes binding of actin and myosin. But in smooth muscle, calcium will bind to calmodulin. In smooth muscle, there is no troponin. So excitation is electrochemical event occurring at the membrane. This is followed by mechanical event, which is contraction of the muscle. This excitation-contraction coupling is termed electromechanical coupling. Calcium permeates from extracellular into intracellular space of smooth muscle that causes contraction. There is another excitation mechanism in smooth muscle, which is pharmacomechanical coupling. In this coupling, there is no change in membrane potential. In electromechanical, there is changes in membrane potential, but in pharmacomechanical, there is no change in membrane potential. But instead, receptors are activated by drugs or hormones. And the source of calcium is from sarcoplasmic reticulum. For contraction to occur, there needs to be calcium ions released. The source of calcium can be either from outside of the cells, which is from the ECF or caveolae, or from in the cells, which is from the sarcoplasmic reticulum. There are basically three mechanisms for increasing calcium ion concentration in smooth muscle cells. One of the mechanisms is through the voltage-gated calcium channels. When membrane depolarize, it will generate action potential, causing this channel to open. So opening of this channel will allow calcium from the ECF to flow into the cell down its electrochemical gradient. There are two types of calcium channels that may be activated by hormone or neurotransmitters. One of this channel is ligand-gated calcium channel. This channel is not regulated by changes in membrane potential, but by receptor-mediated events. Various hormones and neurotransmitters interact with specific receptors. This receptor binds to the calcium channel via G protein. When hormone or neurotransmitter bind to this receptor, the channel will be open, causing influx of calcium from ECF or caveolae into the cell. Another type of channel that is also activated by hormone or neurotransmitter is IP3-gated calcium channel. IP3 is inositol triphosphate. Hormones or neurotransmitters interact with the receptor. For example, norepinephrine, which is a hormone, will bind to alpha-1 receptor. These receptors are coupled to phospholipase via G protein. Phospholipase catalyzes the hydrolysis of PIP2 to IP3. IP3 will then open calcium release channels on the sarcoplasmic reticulum. So calcium will be released from the sarcoplasmic reticulum into the ICF. So these are the three mechanisms for increasing calcium concentration in smooth muscle cell. So when intracellular calcium concentration is increased, the calcium will bind to calmodulin, CAM. Remember that in skeletal muscle, calcium binds to troponin, but in smooth muscle, calcium binds to calmodulin. So when calcium binds to calmodulin, they will form calcium calmodulin complex. The calcium calmodulin complex will then activate myosin light chain kinase, MLCK. Inactive MLCK to active MLCK. The MLCK will phosphorylate myosin light chain in myosin head, and this will increase myosin ATPase activity. So, meaning that the myosin heads will be energized with this ATP. This will enable myosin to bind to actin, or in other terms, myosin will form cross bridges with the actin. Cross bridge is when myosin slide along actin filament. So cross bridges or myosin binding to actin will result in smooth muscle contraction. On the other hand, when calcium concentration in the cytosol is low, the calcium can either be pumped out of the cell back into the ECF, or it can be stored back into the sarcoplasmic reticulum. This is done by calcium pump. So when this occurs, when calcium concentration is low, the calcium will unbind with the calmodulin. Because of this, myosin light chain kinase activity will decrease. At the same time, myosin light chain phosphatase (MLCP) will be activated. MLCP will dephosphorylate MLC, or it will remove phosphate from the myosin head. So this will inhibit myosin ATPase. So by this point, there will be no ATP to energize the myosin head. So myosin will not bind to actin. And this will result in smooth muscle relaxation. In smooth muscle contraction, there is a mechanism called latch bridge mechanism. The latch bridge mechanism is when the myosin head remains attached to the actin for some time, even after the level of calcium in the cytosol is low. We learned previously that when calcium concentration in the cytosol is low, myosin head will detach from actin. But in smooth muscle, that's not the case because there is this latch bridge mechanism. There is no latch bridge mechanism in skeletal muscle. Latch bridge occurs because the dephosphorylated MLC has a very low affinity for the next ATP. So, once the smooth muscle is contracting, the affinity for the next ATP is low, but at the same time it remains contracting without the need for the next ATP, meaning myosin head remains attached to actin even without ATP. So, this is termed latch bridge. The importance of the latch bridge mechanism is it produces sustained contraction or prolonged contraction for hours with little use of energy. This is mainly important to maintain a steady pressure. For example, in smooth muscle of the blood vessel, steady pressure can be maintained on the blood, whereas in the GI tract, steady pressure can be maintained on the GI contents. Another important characteristic of smooth muscle, especially visceral smooth muscle of hollow organs, is its plasticity. Plasticity of smooth muscle is also known as stress relaxation response. Smooth muscle has the ability to return to its original force of contraction after it has been elongated or shortened. For example, in urinary bladder, imagine this is your urinary bladder. When there is an increase in fluid volume in the bladder, smooth muscle fibers will be stretched. So this will cause the bladder to contract, and tension or pressure in the bladder will increase. However, over the next few seconds, the pressure will decrease. Even though the bladder wall continues to stretch, the pressure returns back to the original level. Then, when there is further increase in the fluid volume, the same effect will occur again. So this process is called stress relaxation response. This is important for hollow organs to maintain the same amount of pressure in the lumen even though there is changes in volume. Now back to the smooth muscle of the GI tract. There are two components of the GIT neural control, which are intrinsic and extrinsic control. The intrinsic component is called the enteric nervous system, ENS, while the extrinsic control is by the autonomic nervous system, which are sympathetic and parasympathetic. The enteric nervous system, or ENS, is a set of intrinsic nerve, which is also called the brain of gut. It is considered as the brain because it is the one that regulates the GIT function even in absence of extrinsic innervations. Meaning, even without extrinsic control, either sympathetic or parasympathetic, these intrinsic nerve control the GIT function. Neurons of the ENS extend from esophagus to anus. The neurons are composed mainly of two plexuses, which are mesenteric plexus or Auerbach plexus that controls GIT motility, and submucosal plexus or Meissner plexus that controls GIT secretions. Mesenteric plexus or Auerbach plexus is located between longitudinal and circular muscle layers. So this is the intestinal wall. Epithelial layer situated closest to the lumen. Underneath of epithelial layer is lamina propria, muscularis mucosa, submucosal layer, circular muscle, and longitudinal muscle layer. So notice the black line with the dot. Here is the mesenteric plexus. So the stimulation of this plexus will increase velocity of conduction, causing more rapid movement of peristaltic waves. The increase in peristaltic wave will subsequently lead to increase in contractions tone, intensity, and rate of contraction of the GIT wall. Aside from that, the stimulation of this plexus will also cause inhibition of pyloric sphincter and ileocecal sphincter. So overall, the main function of this plexus is to control motility of the GI tract. Submucosal plexus or Meissner plexus lies in the submucosa of the GIT wall, which is in between the submucosal layer and circular muscle layer. So these plexus control the function within the inner wall of each minute segment of intestine. So these functions include local intestinal secretions, absorptions, and contraction of submucosal muscle. The stimulation of these plexus will cause the infolding of stomach mucosa. This is the summary of the intrinsic nervous control of the GI tract. It consists of two plexuses, mesenteric and submucosal plexus. There are motor neurons connecting mesenteric plexus to the longitudinal and circular smooth muscle layers of the muscularis. So when mesenteric plexus is stimulated, it will send signals to the muscles to contract. From submucosal plexus, there is motor neurons to the mucosal epithelium. When submucosal plexus is stimulated, it will send signals to increase secretions, absorption, and contractions of the mucosa. The mesenteric and submucosal plexus are interconnected by interneuron. Both plexuses receive signals from the autonomic and central nervous system by sensory neurons. Apart from intrinsic control, the GI tract is also controlled by extrinsic stimulation, which are sympathetic and parasympathetic. Parasympathetic innervation to the GI tract is supplied by vagus nerve or pelvic nerve. The vagus nerve innervates upper GIT, such as esophagus, stomach, pancreas, and small intestine, whereas pelvic nerve innervates lower GIT, such as the external anal canal. The postganglionic neurons of GIT parasympathetic system is mainly in the mesenteric and submucosal plexus. So this is the postganglionic neurons. The neurotransmitter is mainly acetylcholine. The effect of parasympathetic stimulation is to increase activity of the ENS. For sympathetic system, the preganglionic fibers are short, while the synapse in ganglia is located outside of the GIT. The neurotransmitters is mainly norepinephrine and a small amount of epinephrine. Sympathetic stimulation usually inhibit GIT activity, and this is done in two ways: either direct effect of norepinephrine on the smooth muscle of the GIT, except mucosal muscle, which it excites. The rest of smooth muscle in the GIT, sympathetic stimulation will inhibit the activity, but for mucosal muscle, it will excite. Sympathetic stimulation may also inhibit GIT activity by the inhibitory effect of norepinephrine on all ENS neurons. This is actually the major effect of sympathetic stimulation. So either the norepinephrine inhibit the GIT muscle directly, or it inhibit the ENS neurons, where the inhibition of the ENS neuron will cause inhibition of the GIT activity. So in summary, there are two neural control of the GIT smooth muscle: extrinsic control, or also called long reflexes, and intrinsic control, or short reflexes. So this is the gastrointestinal wall, which is the site of short reflexes. When there is stimulus, which is substance in the lumen, it will be detected by either chemoreceptors, osmoreceptors, or mechanoreceptors that are located in the wall of the GIT. Signals will be sent to the myenteric or submucosal plexus, where these plexuses will cause either the smooth muscle to increase in contraction, or the gland to increase secretions to ease the digestion process. The receptors will also send impulse to the central nervous system, which will then also stimulate the plexus. The stimulus to the CNS may also come from sight, smell, taste, or thought of food. So meaning even without substance in the lumen, the plexus may also be stimulated. Now, let us look into the electrical activity of GIT smooth muscle. The electrical activity found in the GIT are slow waves, or also called basic electrical rhythm, and the spike potentials, or action potential. Slow waves, or basic electrical rhythm, BER, are spontaneous rhythmic fluctuation of the membrane potential from -40 to -50 millivolt. So this is your membrane potential in millivolts. This is durations in seconds. So this is slow waves, the rhythmic fluctuation of membrane potential between -40 to -50. With some reference say -50 to -60 millivolt, but it depends on your reference. So it's about -40 to -60 millivolt. Slow wave consists of depolarization and repolarization without muscle contraction. During slow wave, there are influx of sodium into the cell as well as potassium efflux out of the cells. Sodium influx will cause depolarization, while potassium efflux will cause repolarization. Despite this depolarization and repolarization, there is no muscle contraction. Slow wave occur all along the GIT except in esophagus and proximal part of stomach. It originates in interstitial cells of Kegel, which is mesenchymal cells located within the muscle layers of the GI tract. So these interstitial cells of Kegel serve as electrical pacemaker that creates the bioelectrical slow wave potential. The interstitial cells of Kegel are located at the distal part of stomach and small intestine, in the saccular muscle near the myenteric plexus, and in the colon in the saccular muscle near submucosal plexus. The frequency of slow wave, which is about 3 to 12 per minute, will determine the rhythm of GIT contraction. Spike potentials or action potential occur when membrane potential becomes less negative than -40, meaning the potential becomes more positive. This is because there is calcium influx into the cell, together with small amount of sodium via calcium-sodium channels. Remember that increase in intracellular calcium will cause muscle contraction. So when there is action potential, there will be smooth muscle contraction. Higher depolarization or high amount of calcium in the cytosol causes a higher number of spike potential, as well as stronger contraction. One interesting thing to note about action potential in the smooth muscle of GI tract is that its action potential can last longer than action potential in the nerves. There are factors that may increase or decrease the number of spike potential and contraction strength of the GIT smooth muscle. Factors that increase the number of spike potential and strength of contraction, but in other words, it should be something that may depolarize the membrane. So these include parasympathetic stimulation by acetylcholine, hormone gastrin, motility, and stretch. Well, stretch, when there is substance or food in the lumen of the GI tract, the smooth muscle will be stretched. So this will cause membrane depolarization and subsequently will stimulate action potential and cause muscle contraction. So these are some of the factors that may increase the spike potential. On the other hand, factors that decrease the number of spike potential and contraction strength, or in other words, it causes membrane hyperpolarization, the membrane potential becomes more negative. So these factors include sympathetic stimulation, which is by norepinephrine, hormone secretin, and gastric inhibitory peptide. So this is a summary of the electrical activity of GIT smooth muscle. Slow wave, which is depolarization and repolarization without muscle contraction. Membrane potential is about -40 to -60 millivolts. So this is slow wave. During resting, stimulation either by stretch, acetylcholine, which is a parasympathetic neurotransmitter, gastrin or motility will cause influx of calcium, which will cause membrane depolarization. The potential will become more positive than -40 millivolts, and this will result in action potential and muscle contraction. Stimulation by norepinephrine or sympathetics, secretin or gastric inhibitory peptide will cause hyperpolarization. There will be no muscle contraction or a weak contraction. So that's all for this topic, Introduction to GI Function and Physiology of Smooth Muscle. If you have any question regarding this topic, please email me at khairino@usm.my. Thank you.